How do genetics affect seronegative raw onset? When we talk about seronegative rheumatoid arthritis, or SNRA, it's fascinating to see how genetics play a role in its onset. Unlike seropositive rheumatoid arthritis, which has its own set of genetic markers, SNRA is influenced by different genetic factors. Research suggests that the heritability of rheumatoid arthritis overall is around 60%, meaning genetics do have a significant impact. In the case of SNRA, one of the key genetic markers is the HLA-B0, 8 and DRB103 haplotype. This is quite different from seropositive rheumatoid arthritis, where the classical HLA-DRB1, 04 and 10 alleles are more commonly associated. This difference indicates that the major histocompatibility complex genes, which are crucial for how our immune system presents antigens, are not as central in SNRA as they are in seropositive cases. Instead, it appears that SNRA is more closely linked to genetic variations that affect our innate immune responses. For example, mutations in non-HLA genes such as ANCLIBL have been identified as associated with SNRA through genome-wide association studies. These genes are not typically linked to seropositive rheumatoid arthritis, highlighting a distinct genetic profile for SNRA. Moreover, genes that play a role in immune signaling pathways, like those coding for Janus kinase and signal transducer and activator of transcription proteins, are more relevant to seropositive rheumatoid arthritis. This suggests that the mechanisms behind SNRA may rely more on innate immunity rather than the adaptive immune system's pathways. Interestingly, studies on microRNA expression have also shown differences between SNRA and seropositive rheumatoid arthritis. Certain microRNAs, such as Hasmir360-5P and Hasmir7083P, are uniquely upregulated in SNRA. This points to a distinct genetic regulation that influences how the disease starts and progresses. In summary, genetics affect the onset of seronegative rheumatoid arthritis through a unique set of markers that differ from those in seropositive rheumatoid arthritis. The involvement of specific HLA haplotypes, non-HLA gene variants, and the emphasis on innate immunity mechanisms all contribute to this understanding. It's important to recognize that while genetics play a crucial role, environmental factors and how genes interact with these factors are also significant in the development of SNRA. Therefore, genetics alone do not dictate whether someone will develop this condition. Consulting healthcare professionals for a thorough evaluation remains essential for diagnosis and management. Understanding these genetic influences not only helps in recognizing SNRA as a distinct subtype of rheumatoid arthritis but also supports ongoing research into personalized treatment approaches for those affected.